Welcome, my name is Cédric Notre-Dame. I'm a researcher and my main topics of investigation revolve around genomics and the development of algorithmic methods for the comparison of biological sequences using evolution. My work involves biology and computer science. I'm also the chief editor of NAR, Bioinformatics and Genomics, a new scientific journal entirely dedicated to these topics. I have been teaching bioinformatics for over 20 years and I have produced a lot of introductory material, including Bioinformatics for Dummies, yes. Over the next 4 weeks, I will be introducing for you the main notions of bioinformatics. During the first session, I will first give a broad definition of bioinformatics and explain how it fits into modern biology. In the next lecture, I will then remind you of basic principles of molecular biology and how all the knowledge it produces can be processed with bioinformatics methods. Later on, we will see what are the tools that make it possible to work with this data, how they work, and more importantly, how they can be used for specific research tasks and clinical analysis. Bioinformatics is a very general term coined by Paula Hogeweg in 1970, though the original sense has drifted quite a bit. Today, it relates to a domain of science dedicated to the computational handling of biological data. It means we use computers to do biology. Bioinformatics is used to store data, to analyze and to compare it. This analysis results in experimental models that allow predictions. Bioinformatics deals with managing the production of data, especially large-scale data like sequencing. We need bioinformatics because the machine generates too much data for a human to handle it. Bioinformatics techniques are used to store and organize this data. This is an important question because data must be stored so that it does not occupy too much space. But it must also be stored in ways that allow analysis to be performed efficiently. For instance, if you compress everything to the maximum, you will need little space. But analyzing your data will become extremely slow, or it will require a lot of computational power. This is not an easy task, and it requires frequent adaptation because of rapid technological evolutions. Over these last years, the amount of data produced by biology has doubled every year almost. This is faster, much faster than the growth of computing capacity that double every year and a half. What it means is that if every year you spend the same amount of money in producing data, you will have to pay more money to store it, or you will have to be better at storing it. You will soon see that this dilemma between spending more money or being smarter is ubiquitous in bioinformatics. And since money is limited, we have no choice but getting smarter. With data ready on your desk, bioinformatics then has to deal with its analysis. For this purpose, programs need to be developed that allow extracting the signal contained in the data. For instance, if you sequence a genome, the first analysis you will need to do is the assembly of all the sequences you have collected into a complete genome. This task is named genome assembly, and it constitutes a very common task in genomics. A very large number of analyses can be carried out by bioinformatics. They range from simple tasks like counting frequencies of nucleotide up to very sophisticated analyses involving functional genomics, protein interactions, and regulatory networks. Over the years, a very large number of such methods have been designed and published in the scientific literature. Some are among the most cited publications ever, like the Cluster W paper that describes a method allowing the simultaneous comparison of many sequences, a technique that is often used to predict the clinical severity of a mutation. All these methods are usually available freely and sometimes even available online, though it should also be possible for you to install them on your own computer and to run them locally. This means that to use them you should send your data, usually by pasting it onto a web browser, and the computation takes place in the lab that has developed the method. It's like a gift they make you. Every year, the journal NAR publishes a list of such web servers. Some of the most commonly used methods in bioinformatics involve data comparison, comparing two pieces of data. The reason is very simple. If you sequence a gene coding for a protein, it tells you nothing about the function of this protein. It only tells you about the sequence of the protein. Unfortunately, it can cost millions of euros to experimentally establish the function of a protein. The good news is that if you can compare the sequence of your protein with other proteins whose function has been experimentally characterized, if you find one, then it can save you a lot of time and money. Of course, comparing protein sequences is not as simple as comparing words, and it requires very specific methods. But given the benefits of this comparison, it is easy to understand why bioinformaticians use them so much. Thanks to these sequence comparison methods, it is possible to save money on everything that is expensive in experimental biology. I often show this graph. I call it the money graph because it shows how comparisons make it possible to replace costly data like structure or function, or even available data like origin, with relatively cheap data like sequence. Of course, in order to make most of this comparison, we need reference data sets. That is to say, sequences whose properties have been experimentally established by others. This defines another very important aspect of bioinformatics: the development, maintenance, and access of reference databases. These databases may contain anything that can be useful to scientists, from publications and keywords to sequences, molecules, and even experiment summaries. An important aspect of these databases is that they have been organized so as to be usable by both human and computers. In order to be usable by computers, these databases use very strict formats and controlled vocabularies known as ontologies. When these things work well, and they don't always do, the databases become interoperable and data can be collected and combined across different complementary databases. For instance, the following image is a snapshot of a Swissprot entry, a database summarizing everything known on a given protein and linking to all the other databases. It is important to insist that bioinformatics analyses are not different from normal biological analyses. They involve measuring quantities, comparing them, and drawing conclusions. The main difference between wet lab biology and bioinformatics is that rather than being examined by eye, the data is examined by a computer, and the quantities are estimated on millions, sometimes billions of experimental measures. In theory, this makes the analysis more robust and less sensitive to experimental fluctuation. But it also means that the final analysis is often a statistical analysis. With so much data, almost everything you can think of can be found. So the question is not if such and such signal is or is not in the data. The question is my observation statistically significant? And this is where we need to be careful. With the wrong statistics, the best data can give you the worst answer. For instance, imagine you measure a property on many different genomes. The genome of patient A gives 20, and the average is, for instance, 10. If you assume your data is normally distributed, 20 is a lot, and there is something statistically significant in your patient. But if your data is extremely distributed, then the probability of getting 20 by chance can be much higher, and your patient is not so special. How will you know how your data is distributed? Well, that is simple. You will have to use statistical techniques to model it. It is only with the right modeling that you will be able to assign meaningful p-values to your data. With big data, this is not a simple task. Last but not least, bear in mind that there are two main categories of bioinformatics analysis: data integration and prediction. Data integration is what happens when we collect large amounts of data and consolidate it into a model. For instance, if you sequence a genome, you will use a genome assembly procedure to consolidate all your sequences into one unique model, the genome sequence. When doing so, all your experimental observations, the sequences, may not be compatible because of sequencing errors, for instance. But the final genome will not be a prediction. It will be a model integrating all your experimental data and allowing you to objectively measure the level of compatibility between this genome and the smaller sequences. If you have no more information, you will then use bioinformatics to predict the genes in your genome. The gene models you obtain will not be supported by experimental data. They will only be prediction. It is very important to make a distinction between an experimental model that does not need to be validated and a prediction that will need an experimental validation at some point. So, what we have learned so far is that bioinformatics is now generating huge amounts of data. In order to be properly analyzed, this data will require computing power. This data can be integrated to produce models, and it can also be used to make predictions. The most common predictions in bioinformatics are based on sequence comparison. In order to determine if a sequence comparison or any bioinformatics measure is biologically significant, we use statistical inference. In the next lecture, I will refresh your memory with a bit of molecular biology, and we will see how bioinformatics methods can help making sense of large amounts of data.